Welcome back. Today we're going to be looking at the structure of the cell. Don't forget to subscribe if you are a new student on this platform. You'll be receiving more and free videos. Without wasting time, let's get started. When you say cells, cells are building blocks. It means that small boxes. In actual sense, it looks like small boxes. So when you say cell in science, they are building blocks. They're small boxes, building boxes of life. So the building blocks of life, we call them cells. So we say that cells are basic unit of life. When we say cell, in this chapter we're going to be looking at the cellular level of organization of living things. The atoms, we'll be looking at the molecules, we saw the molecules, so now we are on the cells. So cells, when you join them together, they form what you call tissues. When you have different tissues performing a specific function, they form what you call organ. They must interact. An example of organ is the heart, and then you have the lungs. So they have different functions. The heart is for pumping blood, and then lungs are for taking in and taking out air. So now they must work together to form what you call a system, because a system there must be different organs. Then these systems, when they join up together, they form what you call the organ. Let's look at the molecular makeup of cells. The composition of cells. That's what we call the molecular makeup of cells. The composition of cells, the molecules which make up cells. But we say that cells are the basic structure and the functional units of living organisms. If cells are the basic structure and functional units of living organisms, then it means that without them we cannot have living organisms. If you look at the word cell, it was first discovered in 17th century by Robert Hooke. And then because cells are very small, so you cannot see them with your naked eyes. So what do you need? You need something which can help you in viewing them. So we call them microscope. So you need what called the microscopic techniques so that you can be able to view the cells. So we will see all these things in detail as we continue with our lessons. Let's look at the microscope. Since we have said that cells, we cannot see them with the naked eyes. You cannot see them without any aid, anything helping the eyes. Therefore, we need something which can aid us to see. So that's why we came up with the word microscopy. Micro means something very small, and then scope means to view. So an instrument which can be used to view small organisms. For example, when you say telescope, tel means land, scope means view. So whenever I say scope, it means something to view. So microscope is an instrument, or is a small machine which is used to view small organisms, so that it tries to magnify it so that you can see it clearly. So the microscope was not just, it did not just came up just like that. It had different phases in which it developed. So it started with what you call the glass lenses, or what you call the hand lenses. These glass lenses, it looks like that. So it's just, we call sometimes it a hand lens because we use the hand to view. It's just one lens. So it was discovered, and then they were able to see at least smaller organisms. The hand lens helped in viewing these. These small organisms. It was discovered by this gentleman called Anton. Anton is a Dutch biologist and use this so that can see the blood cells. So the blood cells were able to be seen, at least the outside features, uh, but it could not view everything inside the blood cell. So because of that, then we have to combine these lenses so that you form another microscope which can have a bigger magnification. So if you combine two or more, you form what called a compound microscope. Therefore, the other one is a simple microscope, while when you combine many lenses, you form what called a compound microscope. For example, a dissecting microscope is an example of a compound microscope. It can view images in 3 dimensions. It can view images without killing them, and it helps us in keeping the species. However, this microscope can also go up to, although it can go up to that, but it cannot view all the features still. There are some small microorganisms, there are some small organisms which cannot be viewed. It cannot be viewed. It requires a bigger magnification. So we require another microscope or another kind or a version of microscope. So, but before we go to that big microscope, let's look at this light microscope. Light microscope is the way you hear it. It requires light. So we say that compound microscope have more than one lens. We will see this one in details as we discuss about magnification. How do you calculate magnification in detail? We will have a special class for this so that at least we can remember how these things are done. So now we are saying that the eyepiece lens, and then this is another lens. These are lenses. This is a lens, this is a lens, this is a lens. The longer the lens, the bigger the magnification. It means that this one has a lower magnification compared to this. The longer the lens, the bigger the magnification, according to the light microscope. So light microscope has different parts. This one is it helps in reflecting light. This one is just to hold. Sometimes you call it the arm. This one is to focus. This and this are very important in focusing. When you are viewing, you first use this, and then after that you use this. So if you are using this, then it means that you are trying to bring up the focus. That's why we call it coarse adjustment. Coarse. At least if you get the focus somewhere, but it may not be clearly viewed. The object may not be clearly viewed. So what you do, you bring what you call the fine adjustment. You do the adjustment finely, and then you have this objective lens. Here you have the first lens. These are the skin lenses. Depending on the magnification you want to use to view the object, therefore, if you want a bigger magnification, then you have to increase the objective lenses. Stage is where we put the specimen which we want to view, and then we have these other parts. These ones we call them clips. So when you put a slide with the object you want to view, the clips hold the slide, so that it does not move up and down or left and right. Then you have this one, we call it the base. Diaphragm allows the amount of light to enter. It's like opening the door. You can open the door widely, or you can open door halfway, or you can just open with just a small slit. So when you use a diaphragm helps in opening the door. If it is you have opened it widely, then it means that you are able to see to receive a lot of air or a lot of light coming into the house. But if you open it in a small way, then it automatically won't be able to get enough. But that light which is entering might be too much in terms of sharpness, because it strikes the door, and then all of it tries to squeeze itself into a small slit. So basically that's so we will see the magnification and how do you calculate the magnification in detail. We will see that as we will organize a class specifically for that, but we will have at least some few ideas about it. Then you have, we say that this one cannot magnify to the a specific magnification we need. Therefore, we had to, the scientists had to invent another kind of microscope which called the electron microscope. This one uses electrons, the way you hear it, uses the electrons to view the objects. So it has its magnification is too big and it can only see objects when are dead. So with the development of the electron microscope, the microscopic details of organelles such as mitochondria, chloroplast, they are able to be seen. Yes, it uses electrons to see. So because of the beam of electrons which they use, therefore it requires the organisms to be dead. It cannot view live organisms. Electron microscope is big, as you see it here in the picture. You need something which is bigger or a bigger space, and you cannot. It is not portable. You cannot just take it from one point to another. You can only see the objects using the screen. The only way you can see the objects is by using the screen, and then you're supposed to install it in a specific look at the structure and the function of the cell. But before you go to the structure and function, let's look at the cell theory. Where did the cells come from? Which specific features do cells have? Cell theory was developed in 1839. So the science is being traced back that way. Yes. So who discovered Skilding and Stewart discovered the structure of the cell. So they brought up the properties of the cell. They say that, number one, all living things are made up of cells. So all living organisms are made up of cells. They're made up of small blocks. They're made up of small blocks. It's like saying that all buildings are made up of blocks. So these blocks are the one which make up a building. Without the blocks, you cannot have a building. So all building, whether it's which shape or which shape, they have the same kind of material, but the way how we use this material brings about a different shape. All organisms are made up of building blocks called cells. Number two is new cells are created by old cells. It means that you have the old cell and then you get a new cell. It means that by reproduction. And then you're saying that cells are basic building blocks of life. As I say that all living organisms, they have cells. So they are the building blocks. They one cell, another cell, another cell, another cell, then they form what you call an organism. So without cells, no life. Then the modern understanding of cell theory extends the concept of the original cell theory to include the following. The activity of organisms depends on the total activity of independent cells. Each cell is performing a specific function. So all the activities, when they join them together, then you bring about the activity of the whole organism. So the cells in the heart, the cells in the lungs, the cells in the brain, they have different functions they're performing, and each cell is performing its function. So when you combine all those activities, you form the activity of the organism. If the heart stops, it means that the cells of the heart cannot perform anymore, then automatically it will affect the whole organism. Therefore, these activities must join together, must be totaled up. Yes, when you total it up, then you bring about the whole organism's activity. Number two is energy flow occurs in the cell through breakdown of carbohydrates by respiration. We will see this in grade 11, that cells cannot perform without energy. That's why we eat, so that we can get enough carbohydrates so that respiration can take place. When respiration takes place, energy is being released. So cells contain the information necessary for the creation of new cells. When you find that cells, they have DNA. This DNA is very important in controlling the activities of the cells, and also by allowing the next generation to have the characteristics similar to the previous generation, which we call the heredity. So when you talk about heredity, you are talking about that inheritance of one character or genes from the previous generation. Content of cells from similar species are basically the same. Like if you have humans, you find out that they have the same characteristics whatsoever. Doesn't matter whether you are white, you are an Arab, you are Chinese, you are what, you have the same genetic plan. The genetic plan is the same. So the content you have as a person from, for example, China or Japan, the same content I have in my cells. A person who is coming from Africa, a person who is from Australia, a person who is from America, all that. So all of them, they have the same content. And then we say that you cannot see the cells, but you have to do what you call the calculations of the cell. So let's look at the total magnification and the size. When we talk about total magnification of a microscope, multiply the magnifying, the power of the eyepiece. We saw that the eyepiece, where you put your eye, we saw it on the microscope, then times the power of the objective. Those ones we say that this one is short, this one is longer, this one is the longest. So it depends on which kind of lens you have picked from those lenses. So the one which is on top times the one which is down, then you get to call the total magnification. We will see this in detail. We will have a specific class for this. Then you're saying that formula for calculating actual size of magnified specimen. The actual size of magnified specimen is, you get it as is equal to the size of the image. Now you know the size of the image. You are given a paper, they will say that this is the size of the image. If it is not calculated, they will give you a scale. We will see this in detail. They will give you a scale, then you can use your ruler, and then you try to calculate the size of magnification. Then times the total magnification. Total magnification is how you have calculated it here. The eyepiece power times the objective lens power. Then you divide it by the total magnification. The size of the image, you calculate it from the paper, and if it is not given, and then you get the actual size of the specimen. But if you're using a microscope, the actual size of the specimen must be very small. It means that this number must be very big, and then this one must be very small, so that you bring a very small if you get here a very big size, a very big figure, yet you are using a microscope, then automatically your answer is wrong. It's not might be wrong, it's wrong. So you have to try to prove whether your answer is at least relatively right or wrong by using that idea. Let's look at a small example. The size of the image, count the number of millimeters division that fit across the diameter of the field of view. So it means that what you see. Yes, so you try to calculate the number of millimeter division, and then multiply the number by 1,000, so that we can change it. We can change the millimeter to micrometers. So to change millimeters to micrometers, you need times 1,000, so that if you get a field of view is 3.5, then automatically it's gonna give you multiply it by by 1,000, then it's gonna give you 3,500 micrometers. So whenever when you are calculating, try to convert this into micrometers, or you can also convert it into nanometers. Then calculate the magnification and the size. Calculating magnification and the size using the scale bar. This the scale bar which I talked about, that sometimes the actual size is, you don't know the actual size, but you have the scale bar on the diagram. We're going to see these in details. We are going to have a specific class for that. Then let's look at the actual size. Actual size on the diagram, the size of on the diagram, they always give you this times the number of this the scale bar. So the scale bar, they give you a scale bar pip, the scale bar, so you have to use the scale bar, then divide by the measure the length of the scale bar. So you try to measure it so that it gives you that so let's look at the plant and animal cells. Plant and animal cells.